Good morning. I'm Konstantinos Mamis, a postdoctoral researcher in the University of Washington in the group of Ivana Bozets. And this is a joint work with Ruibo Zhang, a PhD candidate, and Ivana. And I'll be talking about a simple stochastic model for cell population dynamics in colonic crypts, in continuation a bit to the plenary talk that we also heard today. So our motivation is the central question, like how healthy tissues maintain their size via a hierarchy of different cell populations. That's, of course, a question in many tissues in human body, and how this homeostasis is disrupted by the acquisition of driver mutations. And we particularly focus on colonic crypts, which are the building blocks of the colorectal epithelium. And in our formulation of the model, an important aspect is the stochastic character of cell dynamics. And actually, this stochastic character is amply demonstrated by the significant variance observed in cell populations from crypt to crypts. So a bit on the colonic crypts. The main characteristic is that we have a cell hierarchy depending on the proliferation of cells residing within the crypt. And of course, for healthy colonic crypts, we have homeostatic steady state, where the cell proliferation happening in the lower part of the crypt is in equilibrium with the cell turnover happening in the top part of the crypt. In the bottom of the crypt, we have the stem cell niche in the crypt base, where stem cells divide once a day, producing TA cells. Then we have TA cells that are rapidly divided every 12 to 16 hours and migrate upwards towards the intestinal lumen. After some round of divisions, the TA cells lose their proliferation capability and become FT, fully differentiated cells. The fully differentiated cells, after a short lifespan, they undergo apoptosis. So we would like to model this cell dynamics, and the model characteristics should be the model to be simple in its formulation. And we'll see that because of its simplicity, all parameters except for one are directly estimated from the literature. Also, our model is able to model cell dynamics in both healthy and mutated crypts. This meaning that it encapsulates the variability in size for healthy crypts, and also can describe the crypt size increase after acquiring the APC and CRAS mutations, two mutations that appear in early colorectal tumorigenesis. And also a mathematical result is that our model is exactly solvable. That means that we have a simple mathematical description of the crypt homeostasis and also its disruption in early colorectal tumorigenesis. So our model is actually this branching process with S being stem cells, TA transit amplifying, and FT fully differentiated cells. And this branching process models this cell dynamics shown here schematically, that stem cells actually divide asymmetrically with the rate R, and the number of stem cells N0, we assume that it stays constant in all crypts. Then the TA cells can either divide symmetrically with rate lambda, or they can differentiate into FT cells with a rate D. And last, the FT cells can only undergo apoptosis with a rate gamma. The stochasticity of the model is based that all this cell division, differentiation, and apoptosis, while they happen with constant rates, they happen at random times. So we are able to formulate and solve the corresponding Kolmogorov equations analytically for this branching process, but I won't bore you with the details. So from this solution, we can actually derive the exact distributions for the TA FT cells and the total cell populations. And we can also derive closed form formulas for the averages and variances of cell numbers. You can see here the formulas for the TA cell population and also for the FT cell population. Moreover, we also have the probabilistic structure between TA and FT cell from our model solution, thus having the covariance of TA and FT cells in closed form. And by using all the previous results, we are having the average N variance of crypt size in closed form. Thus, we have those results for the total cell population and crypt. The first basic result of our model is a homeostatic condition. So all the previous results that actually result in a finite crypt cell population within the crypt are obtained under this condition that the TA differentiation rate T is larger than the TA division rate lambda. And this is the condition for crypts to maintain a finite cell population. And if this condition is violated, our model predicts an unbounded growth of cell population, which indicates malignancy. So this is a homeostatic condition, and also it's the model prediction of when malignancy happens. And we can also have homeostasis maintained under different apoptosis rates. We can see from the literature that the FD self lifespan varies from less than a day up to 3 days. Thus, apoptosis rate gamma for FD cells is a quite uncertain parameter. So we've seen, we will see results from our model with the TA differentiation rate D being adjusted in order to have a constant crypt cell population around the values that are experimentally observed. So by doing this, we see that the probability distribution of total number of cells per crypt actually is unaffected by the change of apoptosis rates. And this is actually achieved by the adjustment of the TA and FD cell population. And actually, we can see for the lower apoptosis rates, we have a decrease in TA cell population, which is in accordance with observation already made in the literature.
So do you need to like change some other parameter to make that happen, or this just emerges organically from the system?
It emerges organically from the system if we only adjust D. So we assume that the differentiation rate can be adjusted in order to maintain an average, the same average size of cells in crypts. And these are our results for healthy colonic crypts. The histograms here for cell populations are from 49 human biopsies that are reported in Bravo and Axelrod 2014 and Young et al. In 2017. And the purple continuous lines are the distributions predicted by our model. For the model parameter values, we have direct estimation from the literature for the number of stem cells, the division rate, the TA division rate, and the apoptosis rate. While the TA differentiation rate D, we have no direct estimates from the literature, and we adjust it so the average crypt size agrees with experimental data. And we can see that our model results are in agreement with the experimental data, and actually they pass the chi-square goodness of fit test. So these are the first encouraging results. And now we also have our first results for mutant crypts, thus being able to demonstrate that our model predicts the size and composition of crypts in early tumorigenesis, where the acquisition of the APC inactivation and the CRAS activation happens. So for the APC inactivation, we see from the literature that it's sufficient for colorectal adenomas to initiate. And the effect of this mutation on model parameters has been reported that it's a 60% increase in stem cell numbers N0. With this increase in stem cell numbers, our model predicts a 60% increase in all cell populations and crypts. And it has been experimentally observed that we have a proportional increase up to 100% in the TA compartment and total crypt size after the acquisition of APC inactivation. The same procedure can be made for the crass activation, which is often detected in human hyperplastic polyps. And the effect on model parameters of crass activation has been reported to be a 60% increase in stem cell division rates. Under this increased stem cell division rate, our model predicts a 60% increase in the TA and FT cell numbers in crypts. And indeed, in experiments, we see approximately a 65% increase in crypt area in human hyperplastic polyps. Another encouraging result is that by changing for APC mutation and cross activation, respectively, the number of stem cells or the stem cell division rate, our model predicts the same percentage of proliferative cells in both wild type and mutant crypts. And actually, this is something that has been experimentally observed in hyperplastic polyps and adenomas in humans. So just to recap, we proposed here a simple stochastic model for cell populations in crypt that actually can be solved analytically. And the nice result is that we only fit one parameter and by using the average crypt sizes, but this results in good model prediction for the probability distributions for cell population, not just average cell numbers. And this is in agreement with experimental data. By using simple deduction as we did before, we can have a quantification of the effects of driver mutation on cell populations within the crypts. And we also have a really simple and neat condition for homeostasis. And with this violation also, it's our model's criterion for unbounded self-growth number, thus malignancy. These results and many more you can see in our preprint with the same title we have recently published in bioRxiv. And I'm really happy to take any questions on this. Thank you.
Already had a question.
Really, I would love to see the equation sometime, but
of course,
yeah. The question was, so the crypt architecture is also important. Now, in your system, you're not bothering about the architecture, but what it also means is that the cells that are forming your fully differentiated cells are localized closer towards the crypt end, right? And so that means that not all your TA cells are actually going to FD cells at the same rate, because otherwise it would be a mixed system. How would you, I know it doesn't seem to matter in your system, but in physiologically, maybe it does. And I don't know if it fits in.
Yes, I completely agree. So the thought behind this model is actually expanding on compartmental models that are already out there, where they do not take into consideration the spatial formation of the crypts. But the main disadvantage is that most of them are deterministic, not stochastic. So would like to incorporate the stochasticity. Right. I personally would like to do more about incorporating the spatial characteristics, but this would mean that the model won't be as simple as it's now. And we really want this to be simple and give like really simple results in order to be used further in our project in determining the acquisition of mutations, how we can see the mutation acquired in pre-malignant states in colorectal cancer.
Yeah, so I completely agree with going simple. And the fact that the simple model is actually giving you results that look so good makes me wonder, like, if you actually add that complexity, you might be pushing it away from what's already working. And that's where I'm sort of like bothered. Usually, if you add that complexity, it will look better, but you're already predicting the populations the way you should, by considering that not even everybody's doing the same thing when you know that it's not.
Yeah, that's a question like to be like to be studied. Yeah, but it's really interesting.
Thanks.
Thank you.
Thanks, Konstantinos. Two things. First, why did you choose stochastic instead of deterministic? I think you may have answered that already. Second part, I would be interested in discussing a few details about the solution of the model because my knowledge is very limited in this field.
Yeah, so about the stochasticity, as it was experimentally observed, the variation in cell population from crypt to crypt is really high. So the coefficient of variation for total crypt size has been experimentally observed to be around 30%. So it's high. We would like to incorporate that in our model as improvement from the existing compartmental models that are deterministic. Now, on about the whole solution of that, it's the formulation and solution of the Kolmogorov equations. It's done analytically. It has a bit of like computational burden, but yeah, I'm really happy to discuss it. This is also the main mathematical result that we are having, but it's a bit like complicated to show the procedure of the solution in a presentation.
Thank you.
Thank you.
Right, thank you very much.